Okay, this is the Professor, CBMD PhD, and today's Monday, September 18th, 2023. It is 5:02 and 54 seconds, p.m. Eastern Standard Time, and today's research, colon tumors. So let's jump to it here. And blocking proteins can pull the plug on power for colon tumors. Date September 12th, 2023. Source, Virginia Commonwealth University. The scientists discovered a previously unknown interaction between proteins that is responsible for supplying energy to tumor cells and could hold significant implications for the development of future treatments for colon cancer. The potential answer lies within a class of fatty compounds known as ceramides. Ceramides regulate a number of vital cellular functions, and many cancer drugs stimulate ceramide generation to help fend off disease. When that production of ceramide is cut off, cancer cells can survive and grow more efficiently. The scientific investigators, a research team, began extensively screening cells in the laboratory to identify what proteins regularly interact with ceramide-producing proteins to identify potential patterns that could warrant further investigation. The ceramide synthesis, the enzymes responsible for the generation of ceramide, come in a variety pack of 6 different flavors, ceramide synthesis 1 and 6. And through their research, the team observed that the first flavor, ceramide synthesis 1 CERS1, was highly interactive with a particular protein known as heat shock protein 27, HSP27. The heat shock proteins act like chaperones for other proteins to retain their full function. However, an overabundance of them can tip the scales and prevent ceramide synthesis from doing their job. The researchers also noticed that HSP27 activity was higher in many colon cancer cells, while at the same time CERS1 activity was significantly lower, prompting the team to consider if the absence of one was connected to the presence of the other. There is this yin-yang kind of relationship, which gave them the idea to follow it. HSP27 is like the bad guy, holding back the good guy, CERS1. And through this study, they identified a specific biological mechanism through which HSP27 interacted with and inhibited the function of CERS1 and colon cancer cells. And by deliberately blocking the activity of HSP27 in these cells, the researchers confirmed the decreased presence of HSP27 led to the heightened reactivation of CERS1, which in turn forced a reduction of mitochondrial function. The cancer cells rely on mitochondria to have the energy to multiply. Without it, the cancer cells can no longer sustain the amount of energy they need and die. And by blocking the function of these heat shock proteins, they were essentially able to reactivate the cellular hand that can pull the plug on the power source connected to the cancer. And this revelation led to the study to suggest that HSP27 could be a primary target in the creation of therapies for colon cancer that can really go after these protein-protein interactions and precisely reactivate certain enzymes while they don't touch others. And these findings are the results of a decade's worth of research and the team in an area which, they noted, isn't really being explored by anyone else. Although the study showed encouraging results in colon cancer, it helped to broaden the scope of the research to additional solid tumors to which overexpression of HSP27 has been observed, including lung, pancreatic, and prostate cancers. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.